So I think what we all have in common is that we want to understand how the brain exactly works. When studying the brain, we often divide it into different segments using a brain atlas. A classic way to make such a subdivision is through the study of post-mortem brains, revealing local variation in microscale structural properties, such as cytoarchitecture and myeloarchitecture. The emergence of non-invasive imaging methods allowed not only post-mortem studies, but research throughout the lifespan and in health and disease, and also the generation of new cortical mappings based on T1 and T2 weighted imaging, diffusion tensor imaging, and functional MRI. The complementary classic atlases, like the one from Brodmann, for example, are still often used, but more as a reference to the original 2D images of these atlases. But wouldn't it be great to be able to incorporate these kind of atlases in existing processing pipelines as well? My name is Rory, and I'm interested in combining microscale and macroscale neuroscience. I'm a PhD candidate at the VU University in Amsterdam, and today we'll talk about my project, the reconstruction of 6 classic microstructural and functional human brain atlases in common MRI space. These atlases embody different aspects of cortical organization and widen the range of modalities for which cortex-wide neuroimaging atlases are available. They're easy to incorporate into free server MR processing pipelines and are openly available online. So the atlases we digitized in this study are originally constructed by pioneering neuroanatomists and are based on the study of post-mortem brains and represent different aspects of cortical microstructure. Campbell, Smith, and Brodmann based their atlases on cytoarchitecture, so regional variation in neuronal cell size, density, and laminar patterns, and myeloarchitecture, so the distribution of myelinated fibers. Flexig mapped out the temporal and spatial profile of myelogenesis and defined regional boundaries accordingly. The cytoarchitectonic regional atlas of von Economo was previously reconstructed in MRI space, and in this study we digitized von Economo's mapping of 5 larger recurring cortical types and an additional intermediate type, making a more rough subdivision of the brain based on clear spatial differences and similarities. And last but not least, Kleist used Brodmann's cytoarchitectonic atlas as a basis and allocated localized brain functions based on loss of function lesion mappings. To be able to digitize these atlases, we needed the original documentation of the classic atlases based on the modalities summarized in this table. And in addition, we needed means to reconstruct these atlases in MRI space. We used the MNI Colin 27 template brain as a standardized space for manual segmentation of the cortical atlases. The Colin 27 brain was built from 27 T1 weighted scans of the same subject with the purpose of facilitating detailed segmentations of the cortex. Manual segmentation was performed using the TK Surfer tool included in free surface software. TK Surfer allowed us to visualize the cortical surface on the Colin 27 brain. And for navigation through the cortical surface, we made use of the curvature data and the different options included in the TK Surfer tool, such as XYZ axis rotations and the pile, inflated, and white matter surface views. So with the original documentation of the atlases and the standardized MRI space, we started the digitization process. So I will take the left hemisphere and the Campbell atlas as an example, but the segmentation process was the same for all atlases and for both hemispheres. In this image, the left panel represents the original documentation of the classic atlases, and 2 regions are highlighted here. The one in red is already segmented onto the Colin 27 brain in the right panel, and to delineate the orange region, individual landmark points were plotted on the cortical surface, you can see as these white dots, after which these landmark points were connected, and the area was filled and saved as a label file. All brain regions were segmented this way, and each region was saved as a label file. Once all brain regions of a hemisphere were segmented, a color table was created and an annotation file was made. We checked the annotation file thoroughly for any mistakes and corrected them where necessary. So once satisfied with the segmentation, an atlas file was created, after which the digital atlas can be applied to any MRI data. So it's rather easy to apply these atlases to external MRI data and incorporate them in your own processing pipelines. To apply the atlases to MR data, we just needed the atlas file, the corresponding color table, free surfer output of an MR subject, and a few lines of code. In this example, we applied the Campbell Atlas to the left hemisphere of two human connectome project subjects. With the MRI-SCA label function included in FreeSurfer, we then generated an annotation file of the atlas for the specific subjects. To check whether the spatial location of each label was consistent across individuals, we checked the behavior of the labels when applied to random human connectome project subjects. A consideration in this regard may be that the 6 atlases digitized here are based on the investigation of multiple brains and represent an average segmentation. Hence, the regional boundaries in the digital atlases may not one-to-one translate to cortical microstructure of an individual MRI subject. The Campbell's atlas, which you've seen in the previous examples, contains in total 18 regions, which are rather large, like, for example, the parietal region, here highlighted in green. Other atlases, like the one from Brodmann, for example, also contain rather small and oblong regions, like the 3 Brodmann regions 26, 29, and 30, here highlighted in blue. Therefore, we had to make decisions on whether we want to delineate every brain region separately, like Campbell's parietal region, or merge some brain regions together. And in this example, we merged the 3 small and oblong Brodmann areas, 26, 29, and 30. So the consistency of vertex assignment of an individual label across a population is influenced by its size. So a large label will have a larger portion of its vertices in the center, with a very low chance of switching label affiliation. And it's influenced by its shape. So an oblong label has a relatively more border vertices and thus more opportunity to switch labels. Merging brain regions results in a relatively lower ratio of border vertices over non-border vertices, thereby increasing cross-population segmentation confidence. Naturally, we describe all decisions made in our manuscript in which we present these atlases. So to conclude, microstructural properties such as the number, the size and type of cells, electrophysiological properties, the availability of neurotransmitters and their receptors, the interplay between all cells in the cortical column, and the connections of a region to other brain areas underlie the functions a certain brain area is or can be involved in. It may be of importance to consider different aspects of anatomical organization in cortical atlases used in MR processing pipelines, depending on the research questions asked. So applications of the digital atlases may further include the consolidation of neuroimaging findings using different brain atlases, reflecting different aspects of the brain, use better anatomy-guided node selection in connectome analysis, the provision of regions of interest that can be used to test anatomically specified hypotheses, and they offer the opportunity to compare and combine in vivo imaging-based atlases with microscale information. These digital reconstructions of microstructural and functional brain atlases may be of added value by widening the range of modalities for which cortex-wide atlases are available, and the 6 digital brain atlases are publicly available online at our website. So hereby I would like to thank you for your attention. If you want to contact me, please feel free to do so. You can virtually drop by my poster, number 1899, or email me at this address. With this project, we provide interesting new digital atlases for the neuroimaging community, hoping to bridge cortical microstructure, physiology, and modern-day neuroimaging. Thank you.
Hi, my name is Ross Markello. I'm a graduate student up at Montreal at McGill University, working in the Network Neuroscience Lab. And I'm really excited today to talk to you about some work we've been doing looking at methodological variability and vibration effects in transcriptomic processing pipelines. So when I refer to transcriptomic data in neuroscience, I'm referring to one of the most well-known data sets, the Allen Human Brain Atlas. If you're unfamiliar with this data set, it consists of 6 postmortem donor brains taken from individuals from 24 to 57 years of age. And the Allen Institute, based out in Seattle, collected 3,700 spatially distinct tissue samples from these individuals' brains. They used 60,000 or so unique microarray probes to index the expression of about 20,000 distinct genes all over the brain. And so to date, it remains the most spatially comprehensive microarray expression in the human brain. And when they released these data, dating back to 2010 with a few updates since then, they made a number of really fantastic web apps for exploring and working with the data, some really fantastic visualizations. And that has garnered a lot of interest in the neuroscience community. And thankfully, it's been used quite widely in neuroscience research. However, Because of this, the number of processing options that researchers have taken to go from the quote unquote raw data provided by the Allen Human Brain Atlas to analysis-ready data has varied quite dramatically across the field. And so last year, a former graduate student with Alex Fornito, Irina Arnautovic, had this fantastic overview in Neuroimage where they detailed the different steps that researchers need to take to go from the data provided by the Allen Human Brain Atlas to data that you can really work with, shown here as a region by gene expression matrix. And so they made some suggestions based on some kind of standards that they set for each of these different steps. So building really heavily on their work, we went a little bit further and enumerated every excruciating choice that a researcher might have to make when trying to process this data in order to use with their analyses. I'm not going to go into depth here for different processing choices, but there's 12 different choices, each of which comes with a number of options. Some of them are just kind of Boolean options. Do you want to use nonlinear MNI coordinates, for example? Others have a number of other options. For example, how do I select which probe I'm going to use to index the gene's microarray expression in the brain? And so all told, these options total into over 165,000 unique processing pipelines that researchers could use to process the data provided by the Allen Human Brain Atlas prior to their analyses. And so what we did is we asked, how do these processing choices impact the outcomes of the analyses that researchers are doing to relate transcriptomic data from the Allen Human Brain Atlas and neuroimaging data? So our analytic plan was first to process the Allen Human Brain Atlas using all 165,000 combinations of processing parameters that we detailed. And then we were going to assess the results from 3 distinct analysis streams. So also last year, from Alex Fornito, the same group, they published a really nice overview of relating transcriptomic and neuroimaging data in Trends in Cognitive Science. And they, taking this region by gene expression matrix that you get from all of these processing pipelines, enumerated 3 different analyses that you could do. The first is you could correlate different brain regions across genes, across the microexpression data, and get a region by region matrix, similar to a functional connectivity matrix or a structural connectivity matrix. Here they call that the correlated gene expression matrix. And you can do basically anything that you can do with the kind of normal functional or structural connectivity matrices that we use in neuroimaging. The next is you could do the inverse. You could take the correlation of different genes across brain regions and get a gene co-expression matrix. And so using this gene-gene matrix, you can look at, say, how genes are organized functionally in the brain or their relationship to, different parameters. And then finally, you could just either select a few different genes and their expression across brain regions or do some sort of data dimensionality reduction in the matrix to get regional gene expression information that you can relate to other neuroimaging data, things like cortical thickness or myelination, and examine how the genetic expression of different genes, or again, of a dimensionally reduced data, relates to those features. So we wanted to assess results from those 3 different analysis streams. And then critically, we wanted to look at the interaction of the different processing parameters that we chose and those different analysis streams to see how variable the results we got were. So in order to process all 165,000 pipelines, we needed some software such that we could do it reproducibly, reliably, and efficiently. And so our lab developed a software that we've called Abygin. It's an open source Python toolbox. You can go to the URL up here to see the documentation or one of the URLs down in the corner. And so this is designed to serve as an all-in-one pipeline for working with Allen Human Brain Atlas data. There's some extensive online user documentation detailing all of the steps that I listed before for processing, what each one does, what your options are, why you should or should probably not use different options. And it's as simple as just running Abygin with an atlas, brain atlas. So this takes about 1 minute to run with the Desikin-Kiliani atlas, but it can accept any volumetric atlas aligned to MNI 152 space. So this is nice in that if a researcher wants to use their own atlas that they've derived or an arbitrary atlas for which process data doesn't exist, they can use Abygin to process it really quickly without having to recode all of the different processing options themselves. So I talked about those 3 analysis streams that we wanted to investigate. So for each one, we did a different analysis. So for the correlated gene expression, this is where we take different brain regions and we correlate the expression across genes to get a region by region matrix. We wanted to reproduce something that's been shown in the literature time and time again, which is that brain regions that are further apart tend to have lower correlated gene expression. We see the same thing with functional connectivity, structural connectivity, and the like. And so this is kind of well established. And so to do that, we took the upper triangle of the correlated gene expression matrix and the upper triangle of a region by region distance matrix, that is just the Euclidean distance between brain regions, and generated a Spearman correlation. For the gene co-expression matrix, that is our gene by gene matrix, we took functional gene communities defined in the Allen Human Brain Atlas's initial publication by Horlich and colleagues in 2012, and we overlaid them onto the gene-gene matrices that we got from all of our pipelines, and we calculated a silhouette score to see how well the functional communities they found fit to all of the different matrices we got. And finally, we wanted to reproduce a correlation that had previously been shown by Joshua Burt and colleagues in a 2018 Nature Neuroscience paper, where they looked at the T1-T2 ratio across neocortex and correlated that with the first principal component of gene expression across the brain. So that is taking the principal component analysis of the region by gene matrix, extracting the first principal component, and correlating and getting a Spearman correlation. So when we do this, we see that the methods that we choose matter. So what I'm going to show here are some distributions of the correlations and the silhouette scores that we got for each of our analyses. And so for the first one, for this distance correlation, we see that we get Spearman correlations ranging from below -0.5 all the way up to almost 0, which is a really large range. And here the distribution is separated by the processing parameter that had the greatest impact on this. So holding all other processing parameters constant, changing just this one thing tended to have a really significant impact. And here, that was the precision with which we were matching different tissue samples to brain regions. The more precise we were, the lower the correlation it was, which is actually what we would expect. For gene co-expression, we see 4 distributions here. And again, the silhouette score here is ranging quite a bit. And these distributions are separated based on the gene normalization method, where we see that not normalizing the gene expression data actually leads to poorer goodness of fit of those functional communities in our gene-gene co-expression matrix. And finally, for our regional gene expression, that is where we're correlating the first principal component of the gene expression matrix with the T1, T2 value across brain regions, we see an incredibly skewed distribution. Here, again, there are actually 4 distributions. This is the same parameter had the greatest impact, gene normalization method. What we see is that the only choice that really had an impact on the results was that if you chose not to normalize the gene expression data, you would get correlations that could be very, very, very low. Then what we looked at was the difference between those distributions that I was just showing. So if we take, for example, in the correlated gene expression, the difference between the red and purple distributions, we get this kind of difference distribution. And we see that some of them are centered on 0, but some of them are quite impactful, suggesting that there's actually an interplay with other processing parameters beyond just that most impactful one. And we see that as well for the other analyses. So the question is, what are the best practices? After kind of examining all 165,000 pipelines, which one is quote unquote best? And unfortunately, there's no right answer. But our suggestions would be to consider the recommendations of Arnakevich et al. In their 2019 neuroimage paper, and consider using Abogen for a practical, easy-to-use implementation of those recommendations. The defaults of our software are the suggestions that they laid out in their analysis. So I just want to thank my lab funding resources and our collaborators, and leave you with this to take home. Thanks so much for your time.
Hello, today I'm going to talk about our prospective diffusion MRI data harmonization approach, which is critically important for the joint analysis of multi-site studies. I have nothing to disclose. Diffusion-weighted magnetic resonance imaging has been actively used in many neuroimaging studies to characterize tissue microstructure in health and disease. However, diffusion MRI signal is sensitive to molecular water motion. Even the scan time of the day or fluid intake might have some effect on the diffusion signal. For example, when same subjects were scanned on the exact same scanner twice, we observed around 2 to 4% intra-subject variability in white matter and up to 10% in gray matter of fractional anisotropy. And the plot shows the test-retest variability on 3 subjects, and two sites. If we look at closer to the first site, we can easily see that the present intrasubject variabilities within site are not significant. On the other hand, the slightest difference in hardware, software, or sequence of the scanners can induce much larger unwanted and nonlinear scanner effects on the diffusion signal, which makes it impossible to naively pool the diffusion MRI data across multiple scanners for joint analysis. For example, when the same subject scanned across two different scanners with the similar sequence, we observe significant difference, around 10% difference in fractional anisotropy in white matter, about 20% in gray matter, and up to 35% difference in CSF areas. When we look at also to the plot again, we can see that the existing scanner differences across two sites now are statistically significant. These two examples clearly acknowledge the common agreement that scanner differences can cause substantial and significant unwanted effects on the diffusion signal. The efforts to remove the unwanted scanner-induced effects from the diffusion MRI data are termed harmonization. A harmonization method can be considered successful if the intersite variability of the harmonized data is close to the intrasubject variability, as well as the harmonization preserves the biological variability of each subject during harmonization. There have been multiple attempts for harmonization of multi-site diffusion MRI data. We can categorize the methods into two. First type of methods are data pooling techniques, which aim to statistically remove the scanner differences from the derived diffusion measures. Second type of approaches remove the scanner-specific differences from the raw diffusion MRI signal, which also includes multiple work of ours. The approaches in this category are applied at the signal level in the early processing pipeline. Therefore, the method allows the use of any diffusion MRI data processing like tractography and model fitting method after harmonization. Depending on the availability of the traveling subjects, two primary approaches have been proposed by our group. First one is retrospective harmonization. It is for use where data has been already acquired without traveling subjects. Therefore, it uses age and sex match controls to learn the scanner differences. I would like to note that our retrospective harmonization code is available online if you're interested in. Second one is prospective harmonization, which includes more ideal and state-of-the-art harmonization techniques, but it requires traveling subjects scanned at each site to create a within subject overlap cohort. Scanning a traveling subject on different scanners should ideally be performed in quick succession to avoid intermediate software upgrades and age-related effects. Although the approaches in retrospective data harmonization can be also used in prospective harmonization, more advanced nonlinear strategies such as deep learning should be preferred for more accurate harmonization. To capture scanner differences, we propose rotation invariant spherical harmonics features using spherical harmonics coefficients. In the figure, you can see the Risch features for the same subject scanned with two scanners. Risch features represents the energy of the signal at different frequencies, and they represent different microstructural tissue properties which are independent of the orientation. In our strategy, we select one scanner as reference, and the other scanner to be harmonized or mapped to the reference is termed as target. So using the traveling subjects, we first aim to learn the nonlinear mapping of the Risch features from target to the reference, and then we use the learn nonlinear mapping to harmonize the unseen scans of target. The learning part is performed using training data, which is a subset of traveling subjects that were scanned with both scanners. And from the diffusion MRI data of the individual scans, we first compute the spherical harmonics representation of the signal, and then the Risch features to create the different aspects of the diffusion signal. Next step is learning the nonlinear mapping using a robust deep learning algorithm. And I will go more details of this on the coming slide. In the learning steps, we first segmented the white matter, gray matter, and CSF tissues using the L0 Risch feature, since L0 Risch features presents the contrast of the three tissue classes, then we constructed cycle generative adversarial network with target Risch features as input and reference Risch features as output with their corresponding segmentation maps. In addition to the adversarial and cycle consistency loss that are typical to cycle GAN networks, we included segmentation loss as a multi-class cross-entropy loss to regularize the generators and preserve the variability of the tissues. And after learning the nonlinear mapping of Risch features from target to reference using our cycle GAN networks, we apply the learn mapping to all subjects in target for harmonization. Here are the steps. First, we create the Risch features. Later, we apply the learn nonlinear mapping to estimate the harmonized Risch features. Then we compute the voxel-wise scale map using harmonized and original Risch features and apply to the spherical harmonics to estimate the harmonized coefficients. In the final step, we reconstruct the harmonized signal using the harmonized spherical harmonics coefficients. We first applied the prospective harmonization method to 16 human connectome project subjects, where the subjects were scanned with both 3 Tesla connectome and Prisma scanners. We harmonized diffusion MRI data of Prisma to connectome. In the table, you can see the comparisons before and after harmonization for 5 diffusion metrics. We computed normalized mean squared errors on 16 subjects with leave and out cross-validation for FA, MD, the average diffusion MRI signal itself, as well as Risch features. You can see that the existing scanner differences were extremely minimized after harmonization and almost came to the intra-subject variability level. In the right figures, you can see the voxel-wise comparisons, which also agree that the scanner-related differences were almost come to the were very close to zero after harmonization. We applied the second experiment on human connectome project 3 Tesla and 7 Tesla scans, and you can see the voxel-wise absolute percentage differences of original 3 Tesla scans for one subject before harmonization, and these differences were extremely minimized after harmonization. So in order to ensure that our prospective harmonization method doesn't change the fiber orientation, we have also done a tractography analysis. First, we perform whole brain tractography using a multi-tensor unscented Kalman filter tractography. Next, we use the white matter language to extract specific anatomical white matter bundles from the whole brain tracks. You can see that white matter query language results for cingulum bundle and cortical spinal tract. Qualitatively, in the figures, you can see that the tracks significantly overlap. For quantitative analysis, we computed the overlap distance between the tracks. In average, we have observed 95% overlap between the tracks after harmonization. To summarize, we presented a deep learning-based and biologically unbiased prospective harmonization method using the traveling subjects and Risch features. The method works at the signal level, and it is model-free, and it is applied early in the preprocessing pipeline to minimize the preprocessing related effects on the harmonization. We also observed that after harmonization, scanner-related differences were very close to the intra-subject variabilities. This is the end of my talk. I would like to thank to my mentors and my collaborators, as well as our fundings which supported this work. Thanks for listening. I would be happy to answer the questions.
Hello everyone, I'm going to present on normative modeling in a multi-scanner neuroimaging dataset, a Bayesian approach to deal with scanner effects. Normative models are well known from a developmental perspective. They are the idea underlying growth charts where height is mapped as a function of age. The core of each normative model are the percentiles that you see here, indicating the normal variation of, in this example, height with age. Deviation from a normal growth trajectory in this example would mean that an individual score lies below the 5th or above the 95th percentile for their age. In recent years, the concept of normative modeling has been transferred to the field of neuroimaging. Instead of height, one could map a brain structure onto age. Machine learning methods can then be used to derive those percentile scores. Once established, this normative model can be used to indicate whether an individual score ranges above or below the normal range of variation for that brain structure. The appeal of normative modeling for the field of neuroimaging is that it allows to capture heterogeneity at an individual level, thus allows to make individual predictions. This goes beyond the approach that is currently used in many neuroimaging studies, which is just comparing group averages. This is made possible by the fact that normative models allow to map not only the mean, but also the variation or noise with the x-axis, so in this example with age. This allows to calculate individual set scores, that is standardized deviations from the norm for each individual, allowing to compare deviations between individuals and between age groups. This is especially relevant for clinical studies, as it has been shown that the variation in those populations is significantly large, and it might not be enough to just calculate group means. Normative models, however, as you need to map a lot of variation, need a lot of data. A fast and cheap way to obtain big neuroimaging datasets is by just sharing data or pooling data across studies. To facilitate this, initiatives like the ENIGMA consortium have been founded. ENIGMA is a collaboration of 50+ research groups where researchers have agreed to work together, share their data, and work and publish on their shared data together. This is great because it allows their members access to big neuroimaging datasets. However, not having been collected in a harmonized way, those types of datasets are often confounded by so-called multi-scanner effects. That means they are biased by the data is biased by where it's coming from or where it was collected. I'm going to show you those effects in an example of the ENIGMA MDD consortium, which is led by Liane Schmal. So what you see here is average cortical thickness derived from a free surface population of 4,500 individuals mapped onto their age. You see that in general, average cortical thickness decreases with age, which is also confirmed by the literature. However, if you color the data based on where it was recorded or where it was collected, you see that there are actually huge side differences in the data. Unfortunately, the machine learning techniques that we use to create those normative models pick up very easily on those side effects and induce huge biases. Thus, in order to harvest the full potential of normative modeling for the field of neuroimaging, we need to find a way to deal with those side effects. As part of my PhD, I've been working on statistical harmonization as a solution. In detail, one of my projects aimed to develop a good model to estimate side effects in a normative modeling framework. The approach I took was a proof of principle approach, and I used a publicly available dataset, the ABI dataset. The ABI dataset is actually an autism dataset, but I used healthy controls to create my models. Most importantly, it's a multi-scanner dataset, so the data was collected at 20 sites. I was working with the free surfer data that comes with this dataset, and I used a normative model with a predictor site to predict this free surfer data. I also ran a train test split approach, so I trained my data on a training set and then tested it in a test set. I compared several different models to find the best way to deal with those side effects, but all my models had a vanilla linear mixture model at their core. So I always tried to predict average cortical thickness by some combination of site, age, and sex. The model differed in the way I modeled site. So I had one model random intercept fixed slope where I allowed a random intercept offset for site. In a model random intercept random slope, I also allowed the slope to vary. Then I had two nonlinear models where I added a Gaussian process or GP for age, allowing to capture nonlinear effects between age and average cortical thickness. My approach was also fully Bayesian, so I used STAN, which is a probabilistic programming language and which uses the method of Hamiltonian Monte Carlo to simulate the posteriors. The Bayesian approach also allows me to specify priors and hyperpriors for each parameter, which leads to better out-of-sample predictions and prevents overfitting. I also get an estimate of uncertainty of prediction for each individual and a general noise estimate. And the Gaussian process for age allows for the estimation of nonlinear effects between age and cortical thickness. Then here's some results. So I compared all my models with respect to commonly used measures of model performance. In detail, I used Pearson correlations between the true and predicted value and the root mean squared error. What you see here is Pearson correlations for average cortical thickness for all the 4 models, separated by train and test set. The picture doesn't look very different, but here are the scores, and you see that the random intercept random slope model with the GP has the highest correlation. The correlation then drops a bit in the test set. You also see that the model random intercept fixed slope has the lowest correlation, and this is also statistically significant. So this model performs worse than the other three. Then I want to show you how my model can make individualized predictions. So what I did here is I took just 2 of the 20 sites of the Abide dataset to show you. You see here, I'll start with the linear model. So here's the random intercept fixed slope model. This is the random intercept random slope model. I have here average cortical thickness mapped onto the age of the participants. The black dots are the true values, so like 1 dot for each individual, their average cortical thickness mapped onto their age, colored by sex. Then you see those box plots here, and those box plots are 4,000 forward predictions per individual that my model created. So for each individual, I get 4,000 data points. You see actually that the box plots overlap quite well with the true values. And so the difference between the two models is that in this model, the slopes are the same because they're not allowed to vary, whereas in this model, the slopes are allowed to vary, so they are different. Then for the nonlinear models, here I also can capture nonlinear associations, which you can also see here. The slopes are curved, the same for this one. Then for correlations, I've already indicated that the correlation for average cortical thickness between the true and predicted value was quite high. I also tested other regions, and you see this here. So it turned out that it didn't work for all the regions equally well. You see here the 4 models, and then M thickness is average cortical thickness, has the highest correlation. So for the training set in this model gives kind of the order of the models, of the regions, and you see that average cortical thickness has the highest correlation, and then it drops for other regions. You also see that it generally is lower in the test set. And the difference between the 4 models here was that this model, the random intercept fixed slope model, or the easiest one, performed worse, and this model performed best. Then for the root again, smaller for the training set, and then more error, a greater error for the test set. Again, this model performed worse, and this model performed best. And this is also my interim conclusion. So I ran an ANOVA, and this ANOVA confirmed that the model with random intercept and fixed slope performs significantly worse than the other ones, and the model with the random intercept, random slope, and Gaussian process performs best. So I conclude that I successfully was able to create a normative model in a data set, a neuroimaging data set with scanner effects. And the next steps will be that we are going to apply the best performing model to a clinical data set. So we want to estimate the deviation of individuals with mental ill health, in particular with depression, under a model trained on healthy controls. And then we want to extend the model, so and adding scanner parameters, because we actually think that the field strength of scanner in use might be driving those side effects. These are the people I would like to thank for the project, and now I'm happy to take your questions. Thank you.
Hi, everyone. It's really a pleasure to be able to present this work today. My name is Carolina Makowski, and I'm at UC San Diego, and today I'll be talking about improving discovery of the genetic architecture of the cerebral cortex. So the quest to understand the human cortex stems back ages, really, and many people are interested in this question. And one of the main reasons for that is because when we look at human ancestors, such as the chimpanzee and even mice, we actually share a lot of our genome with these other species, although you can obviously tell that our brains and many of our traits are very different from mice and chimpanzees. So actually we share 92% and 97, 98% of our genome respectively with the mouse and chimpanzee. But it's hard to argue that our cortex, especially our neocortex, is very unique and underlies many human-specific traits such as higher cognition. So many people have tried to understand what makes our cortex unique in terms of morphology and of course genetic architecture. So 2 of the main phenotypes that people look at when they look at the genetics of cerebral cortex is surface area and cortical thickness. So surface area is in blue here, just constituting the area of the cortex and the inner folds. And cortical thickness in this image here in turquoise is the distance between the inner gray-white matter boundary and the outer peel surface. And the reason why people look at these separately is because it's known that surface area and cortical thickness have different genetic variants. They're largely genetically independent, although they share some features. So very recently, Grasby and colleagues published this elegant study in Science looking exactly at this question of the genetic architecture of the cortex. And using the Cetin-Karayumak atlas, they found 306 variants that were associated with surface area and thickness. So this atlas that they used is 34 regions by lobe, kind of based on anatomical landmarks. But it turns out that it really does matter what atlas you use when you are conducting these studies. So some of our colleagues recently published in Cerebral Cortex trying to better understand the amount of variance that we can explain in surface area when you look at different atlases. So in blue here, where you see Chen area, is actually a genetically based atlas that I'll be discussing more today. And what you can see compared to other atlases, such as the Yao atlas, Cetin-Karayumak, and Glasser, the Chen, or the genetically based atlas, has higher discoverability and higher heritability, meaning that it's explaining more variance in surface area with genetic architecture. So the aims of this study are threefold. The first is to use these genetically informed atlases. The second is to see whether these genetic variants that we're finding generalize from a discovery data set in older adults to a neurodevelopmental cohort, because a lot of these studies have been done in older adults since we have more data there. And the third aim is to better understand these genetic variants and to map them to genes and to biological pathways for interpretability. So for our discovery data set, we are using the UK Biobank. So after filtering out by ancestry to make sure it was a really homogeneous data set, and after quality control, we have nearly 32.5 thousand people with imaging and genetics data, and the mean age is about 64. And for our neurodevelopmental sample, what better sample to use than the Adolescent Brain Cognitive Development, or ABCD study? For this, we use baseline data, and we have about 5,100 9 and 10-year-olds. This is about half of the full baseline sample because, again, we wanted to have a homogeneous sample. So this is predominantly kids with European ancestry. And for the association between genotype and phenotype, we of course turn to the genome-wide association study, but here we're using a tool that was published last year called FAST-GWAS. And the benefits of this tool is it's a mixed effects model, so it can include related individuals, which is of interest when we're looking at UK Biobank and ABCD, which includes related individuals. And finally, the genetically based atlases. So we have two independent atlases here for cortical thickness and surface area. And these atlases were published previously by Chihua Chen in Science and PNAS in 2012 and 2013. And these atlases are derived from fuzzy clustering applied to twin samples from the Vietnam twin study of aging. And you can see here that both atlases have a 12 cluster solution, but they're slightly different. And we have some just general covariates that we have in our GWAS study, but I do want to draw your attention to the fact that we have these global cortical measures that we are covarying or regressing out. So this is mean, thickness, and total surface area. And we found that statistically, these measures are really important to control for because they contribute significantly to our model. And finally, for our follow-up, there are many routes that we could have taken here. And if you come to my poster, I can explain some more work that we're doing here. But for the purposes of this talk, we are mapping genetic variants to genes and gene ontologies from FUMA, this online software. So for our results, starting with area or surface area from UK Biobank, you can see here in the figure with the brain images, the number of hits per region, and this is following the clumping procedure in FUMA. So for those that are not in the genetics world, clumping essentially takes into consideration the correlation between genetic variants or SNPs. So this is really important. And what you can see on the right are the Manhattan plots for each of the regions that are color-coded, and the y-axis is the log 10 p-values, so they're scaled the same way in all of those Manhattan plots. And what should jump out at you is right away the occipital area has the highest number of hits, and you can see there's some other regions in the temporal and inferior parietal that also have quite a high number of hits. And next to each of the Manhattan plots, you can see pie charts that show the functional annotations of our SNPs. And what should jump out at you is the preponderance of sort of turquoise, green, and olive. So these are really non-coding regions, introns, non-coding RNA, intergenic regions, which is probably not surprising, but suggesting that our hits are really enriched in these non-coding regions. So for thickness, very similar to what Grasby and colleagues found, we found less hits for cortical thickness, but still some notable hits. And again, we have the same scale for our y-axis as the figure before. And once again, you see this enrichment for those non-coding regions for our hits. And in total, we have 332 hits as defined by FUMA. But when you actually take into consideration the same parameters that are used in Enigma and clumping in the same way, we actually do have more hits than was presented in that Grasby et al. Enigma paper. So for our generalization to ABCD, the neurodevelopmental cohort, we took two approaches here. So the first was just rerunning the GWAS and then adding ABCD in a meta-analysis approach. And using this approach, we found that about 86% of SNPs still remained GWAS significant after including ABCD in the meta-analysis. So these are the hits per region, and they're largely showing the same pattern as just including UK Biobank in the GWAS model. Another approach we took was to compare effectively effect size estimates. So to do this, we took the beta estimates from the summary statistics of UK Biobank, as well as the beta estimates from the summary statistics of ABCD, and we compared them. So on the x-axis, and what we're plotting here are just the top 500 clumped SNPs from UK Biobank and comparing those beta estimates to those we obtained with ABCD. And for instance, here for the occipital area, we see very, very strong correspondence. And looking across all of our phenotypes, we do see very good positive correspondence between these beta estimates in UK Biobank and ABCD, once again suggesting that many of the hits we're finding in an older adult data set are also holding true in a neurodevelopmental setting. So these are just the Pearson R correlations you see on the right. And finally, to make sense of these genetic variants, we mapped these to genes using FUMA, and then we took forward those genes into a gene ontology database to better understand what biological processes are enriched. And here we actually found for several different regions enrichment for biological processes. These are corrected for multiple comparisons with Bonferroni. And these are the ones that survived, and a lot of them are associated with brain development, not surprising. So for instance, for occipital area, we see processes that are involved in visual system development, like iris morphogenesis, pigmentation, tube morphogenesis. For instance, anteromedial temporal area, you see things like neuron fate commitment, forebrain development. But here we're really defining these GO terms with respect to specific regions. And for cortical thickness, we found fewer enriched GO terms, but there's still some really interesting ones here. So for instance, dorsolateral prefrontal thickness, what really struck me is that we have a term for tangential migration. Typically, when we think of development and brain development, we talk about radial migration of neurons. So tangential migration is sort of a more novel pathway that people are trying to discover more of the molecular mechanisms underlying it. And similarly, medial prefrontal thickness has some interesting terms for cell maintenance as well as cell death. So in conclusion, we are finding that this genetically informed atlas is boosting our discoverability of genetic variants underlying cortical morphology. And we are also finding that these genetic variants contributing to area and thickness in late adulthood seem to be important in childhood as well, which we're showing for the first time with the ABCD dataset. And finally, we are finding novel genetic variants that are shaping different cortical regions involved in conserved developmental pathways. And this is giving us a hint that a lot of these pathways may be conserved, but it might be the regulation of them that are being altered in a human-specific way, which is sort of our future avenues for this research. Thank you very much for your attention. I'd like to give a shout out to everyone that's on this slide, but in particular my mentors, Chi-Hua Chen and Anders Dale, as well as some of the ABCD folks here at UCSD, Terry Jernigan, Chen-Chi Fan, Don Hegler, folks at Oslo for UKB processing, and also Peter Vischer and Jean Yang in Australia for their helping with the project with the statistics and methods. And with that, please feel free to contact me, come by my poster, and happy to take questions in the live Q&A.
Hello, thanks for the organizers for making this happen and for inviting me to present our work. Today I'm going to be talking about some work we did with validating the Allen Human Brain Atlas, which is a spatially comprehensive gene expression atlas that many people have been integrating with their neuroimaging data. So basically, we're going to be talking mostly about data that occurs at this level of scale in terms of the biological hierarchy, and the imaging data that most people relate to this atlas is at the macro scale level of this hierarchy. And so how do we best traverse these levels, and what information at the macro scale is being captured at the micro scale and vice versa? And so the Allen Institute for Brain Science released the Allen Human Brain Atlas about 8 years ago at this point, and it's the most spatially comprehensive atlas of gene expression in the human brain, in the adult human brain. And first studies looked at the heterogeneity across cortex and subcortex, and basically found that there were sets of genes that were more highly expressed in the different areas of the brain. And this spatial heterogeneity was related to cell type composition. And recent work within the last 5 years has begun to integrate these gene expression maps with functional and structural neuroimaging data. This first paper, kind of canonical paper at this point, looking at resting state functional connectivity networks in humans and mice, and relating that network structure to patterns of gene expression across the cortex and subcortex. Some recent work that has extended these data into interpolation of these maps into spatially comprehensive gene expression at the vertex level is shown in the left. And this has provided a really nice full cortex coverage of the gene expression, despite the sampling being done at a coarser grain. And I'll be talking about some of that in relation to the cell types later on. And some other recent work has really focused on standardizing these pipelines for mapping the Allen Institute data to a space that's similar to the functional and structural neuroimaging data that we collect, so that we can properly spatially correlate these gene expression maps with maps of functional connectivity and structure that we derive from the in vivo neuroimaging. And so there's various filtering steps that are shown here in different considerations to account for inter-individual variation across the 6 donors in this data set, and also for problems of spatial autocorrelation and potential spatial mismatching. And cell type composition is, of course, a feature of spatial heterogeneity, but it's also a major feature of temporal heterogeneity and basically differences in trajectories of gene expression across the lifespan. And so this recent paper from the Psych-ENCODE consortium, published, I guess, a year and a half ago at this point, took multiple samples from different postmortem subjects across embryonic to development to adulthood, and basically found very similar patterns, such that there's spatial differences in the temporal components of the lifespan, such that the cortex tends to cluster on its own in terms of trajectories of gene expression overall, whereas subcortex has its own trajectory. And right here, I'm plotting just I'm showing the plots that they made purely for cortex. So within cortex, there's also a spatial heterogeneity that is observable also in the Allen Human Brain Atlas. And critically, although there's kind of this canonical gene expression pattern across development, it's clear that later on in their paper, they break this down into specific cell types using single cell RNA sequencing. And clearly this pattern is quite different depending on which cell class you're looking at. So cellular composition is not only an important component to the spatial dimension, but also the temporal dimension. And so this work really stems from the massive implementation and development of single cell sequencing technologies, spearheaded not purely, but definitely based on major contributions were made based on this one paper from Evan Makowski in 2015. And basically the gist of the single cell sequencing is you capture a single cell in a droplet, nanoliter droplet, and therefore you can barcode it and do the sequencing and then map that back to the type of cell. And the Allen Institute themselves has embarked on single cell sequencing experiments from multiple different cortical and subcortical brain regions, shown up here. And this yields a nice clustering of genes based on cellular composition across the neocortical layers, as well as the different cell types of neurons and non-neuronal cells. And so this technology allows us to potentially deconvolve the bulk tissue data that the Allen Human Brain Atlas is made up of and begin to actually make maps of cell composition or based on cellular type from these single cell sequencing studies. And so what that looks like in practice is basically taking one of the regions of the Allen Institute data set and using information about the different RNA makeup and the gene expression makeup of the different cell classes plotted here in different colors to be able to then project that into the full cortical sets of samples and create absolute expression maps for the different cell types. And so this work was part of a larger project, which I won't go into today, where we did a kind of two-pronged validation of the Allen Institute data by using patient versus control maps of multiple different neurogenetic syndromes where we know the causal genetic link, and relating that back to the genes that we hypothesize to be differentially expressed in those patients. And this secondary bit allowed us, or the integration of the single cell data allowed us to do this secondary validation where we could take some of the genes that we knew to be involved in those different conditions and actually ask, well, whether they were part of cell types, specific cell types. And that helps potentially disambiguate the sources of the neuroanatomical variation in terms of the actual morphometric feature that is differentiating the patients from the controls. And so this is basically just a matrix plotting the expression levels in the Allen Institute data. And effectively, we took kind of a hypothesis-driven approach based off of this recent work from from Yale, where they found that there was a principal gradient of gene expression across all brain-expressed genes. So we took just brain-expressed genes in the Allen Institute data, and we aggregated different cell types across 5 different single-cell sequencing studies. And what I'm plotting above is the average expression for those different cell classes determined by the 5 different studies. And so what you can see is a clear clustering based off of cellular type, regardless of which study called certain gene or classified certain genes as certain cell types. So you can see a neuronal classification up here to the right, the different astroglial and endothelial classifications to the left. And so this decomposition allowed us to then project that information back into the brain. So here we were using a weighted average approach from the gene sets that made up these different canonical classes. And in figures B and C, I'm plotting both a spatial decomposition into 2 dimensions based on these expression profiles, just to show the clustering a bit cleaner, and then projecting those cell classes back into the brain, into these kind of canonical types that you can clearly derive from the original data. And so recently we've taken these parcellated maps and actually put them into the same framework as the paper that I showed initially that was for fully comprehensive at the vertex level. This is work being done with Conrad Wagsteel, who provided these really pretty plots. And you can see that the different cell classes at this finer grain level show really different patterns, which were a bit tough to see at the parcel level. And so, for example, this oligodendrocyte signature looks quite similar to a myeloarchitecture map from the Votes from Fleischig, and the myelin maps T1, T2 ratio from the HCP approach. And so we took a quantitative approach to validating these cell type maps at the parcel level for the project that I was mentioning before. And we found some very interesting signals, mainly that an in vivo marker of myelination derived from magnetization transfer was related to oligodendrocytes as expected, and density estimates from a histological sample using the big brain data set were related to the neuronal subtypes. And so we wanted to validate this a bit further, and so we took 2 different regions that showed a dissociation between the astroglial markers, where we also had associated in situ hybridization data provided by the Allen Institute as well, and was also covering those same spatial locations. And we showed that different astroglial markers, canonical markers, myelin basic protein for oligodendrocytes and GFAP for astrocytes, demonstrated this double dissociation as predicted by our cell type deconvolution approach in the Allen Institute data. And so now we can really begin to potentially make inferences, correlative inferences, as we were doing with the Allen Institute data as a whole. But instead of looking gene by gene, we can look at these gene sets, which make up canonical cell types, which we know contribute to spatial heterogeneity. And so the takeaways from this portion of the project was that we can leverage single cell sequencing technologies which are relatively recent and definitely after the release of the Allen Human Brain Atlas. And we can leverage those results and insights from those studies to be able to translate them back to the bulk data that we're using to spatially correlate to our neuroimaging. And so these gene sets are made available. All of the data from the RNA sequencing studies are also available, so you can do this yourself. And with that, I'd like to thank my collaborators, formerly at the NIH and at the University of Cambridge, and everyone else who helped out with this project. Thank you.